Okay, this is the video for making a bacterial smear. You're going to use this procedure whenever you are going to stain bacteria to look at it under the microscope. Unless otherwise told in a staining procedure that you do not need to make a smear, you will always use this procedure. Procedures differ from book to book, but the procedure that I do here is the procedure we will use. Okay, now I'm going to be using a Bunsen burner. I'm going to be using living bacteria, so I want to make sure I have the proper safety equipment. So I need some gloves. So before you ever light that Bunsen burner, you want your goggles on your eyes. Not on your forehead, on your eyes. Okay, so the first thing we're going to do is make sure we have the correct supplies. We're going to need slides, we're going to need the proper bacteria, the inoculating loop, the Bunsen burner, the striker, water, and a marker. So for your procedures, when you go to make a smear, depending on the staining procedure that you're doing, and depending on, I have a little fly with me, and depending on the what stain you're going to do, you're going to use 2 bacteria, 3 bacteria. It's just going to depend on what you're doing. Now, you have a couple of options when it comes to making your slide. If you're going to be using just 2 bacteria, you can actually put them both on the same slide. If you're going to be using 3, I would use 2 slides. Or you are more than welcome to put 1 bacteria on one slide, another bacteria on another slide. However, you're going to keep it less complicated for yourself. Don't worry about using multiple slides, okay? So to start out with, we're going to be doing 2 bacteria. So what we can do is we can actually put them both on the one slide or put them on 2 separate slides. What I recommend you do is you use a Sharpie marker and you draw circles on the back side of the slide where you're going to be putting the bacteria. So you can do 2 bacteria on one slide, or you could do 1 on one slide and then the other on another slide. The reason that we're drawing the circles is that's going to tell us where to put the bacteria. If you don't draw the circles and you go to use the microscope, you won't be able to find them because you sometimes can't even see it with the naked eye. Okay? The other thing that you need to do is on the back side. Remember, this is the back side, the opposite side of where you're going to put the smear on. You want to label the bacteria. So you can either try to write backwards, or you can write upside down, or you could write it the correct way, but when you flip it over, it'll be backwards. So however you do it, it's completely up to you. Make sure you do not label directly in the circle, because when you put the bacteria on top of it, the marker will be underneath where it is. And when you go to look at under the microscope, you're not going to see anything but marker. So I don't recall what bacteria we're using. I assume we're using like Staph aureus or something, and a bacillus. So I'm just going to label an S and a B. Okay, so whatever bacteria you're using, you label it accordingly. Okay, so there's the one with the 2, or you can use 1 of each kind. Completely up to you. Now that I have them labeled, I'm ready to get started. So the idea of making a smear, there are 2 reasons for making the smear. First of all, you're going to put the bacteria on the slide, and you want the bacteria to dry, and you want them to adhere to the slide, meaning stick to it. Because the next step that you're going to be doing is staining it, and you're going to stain it with a liquid. And when you go to put those liquids on there, if it is not fixed, they're just going to slide right off, so you're not going to have them to see. And that's really frustrating when you go through a 15-minute staining procedure, and you go to look at it under the microscope and it's gone. Okay? The other reason for fixing the smear is to actually kill the bacteria. So you don't want living bacteria swimming around while you're trying to stain them or look at them under the microscope. Of course, it's always safer to have dead bacteria, and also you can keep them indefinitely to look at them at a later time or share them with your lab partners and neighbors in class. So we've talked about how to fix it, what we're going to do. So now we're going to go through the procedure. So I have all of my supplies, hopefully. Sometimes I forget. We'll go ahead and give it a shot. All right, that little fly is just making me crazy. So the first thing we're going to do is light the Bunsen burner. Remember, with a Bunsen burner, you're going to have gas coming out and you're going to have a striker. So the first thing you want to do is make sure your striker works. So do it a couple of times and make sure that you're getting a spark. If you don't get a good spark, you can put pressure on the back of it and push a little bit harder on it to get a greater spark. Okay? Now we're going to turn the gas on. When you turn the gas on, you should actually hear it. If you don't hear it, it's probably not on. Then you're gonna hold the striker right above it, and boom, you've just lit it. Well, maybe the fly will go there. So if you find yourself where you're just going like this and this and this and this and this and it's not working, make sure you got a spark. If you don't have a spark, it's not gonna work. Some strikers are better than others. Do not be afraid of the Bunsen burner. Try not to say, oh, I'm never gonna do the Bunsen burner, you always do it. We end up in class where one person is the official Bunsen burner lighter. That's not what I want. I want you to be able to every single procedure. Another thing is when you go to make the slides, every person makes every slide. It's not that one person in the group does this slide and another person in the group does that slide. I want everybody doing every procedure. I don't want to see the tag team kind of sharing. Now you're more than welcome to help each other, but everybody does their own slides. So now we're going to be taking our bacteria and putting them on the slides. So I'm going to go ahead and use the one that I have 2 of, but like I said, you're more than welcome to use 2 slides for 2 bacteria, 3 slides for 3 bacteria, however you want to do it. Just make sure you label them properly, and you label on the bottom because you don't want to rinse the labels off. Okay? So now we're going to look at which bacteria we're going to use. So I said I was going to use Staph aureus, and I said I was going to use Bacillus megaterium. No, it wasn't Bacillus megaterium. It was a bacillus of some kind. So the other thing in most procedures, and like with today's procedure, is you're going to do it from a liquid and from a solid. So I'm going to show you the procedure of the liquid, and then I'll show you the procedure of the solid. Okay? With a liquid, now remember we don't want to pick them up by their lids and they fall out and break. With the liquid, you can just use the inoculating loop, go directly from the liquid to the slide. With the solids, you're going to have to put some water on first. Otherwise, it's going to be too concentrated and it's not going to spread out on the slide. You want a very minimal amount of bacteria. One of the mistakes that students make is they want to put a big, huge bunch of bacteria on there. Then they end up with a big blob of stained bacteria and they can't see cellular structure. So actually less is better. And if you can barely see it with a naked eye, that means it's perfect. Okay? So we're going to do this one first, and the bacteria is kind of sitting on top. Sometimes you may have it on top, down on the bottom, and you want to give it a little mix. So you just kind of hold the glass, not the lid, and give it a couple of flicks, and you get a little whirlpool going, and that'll mix it up so you get a good sample. Okay? Now we're going to use our sterile technique. We're not going to put the lid down. We're going to use the same technique that we've used in the past with sterile technique. Always practice. One person does it all. Not everybody, each person does their own specimens. So you're going to flame your loop, okay, till it gets red. Once again, we're not going to blow on it. We're not going to touch it. We want it to cool off so that we don't sizzle these guys. We do want them dead on the slide, but if you put it in there where it's hot and you hear that, you're making their cell walls explode, and the purpose of staining is to observe the shape of the cell wall. If you explode the cell walls, then you're not going to see a shape. So that's cooled off probably quite enough. So we're going to take the lid off with our little finger like we did in the past. We're going to run it through the flame just twice. We're going to dip down into it, get a good representative sample, flame our lid, flame our tube, not our lid, put it down, and then we're going to put it in the space that is labeled for that particular bacteria. Okay? You want to rub it around and get a very thin smear, but you want to be gentle as to not break the bacteria up. Okay, now we're going to flame this really good because we're going to be using it for the next bacteria. Now, we want to take a solid and put it onto the slide. In this case, we're going to have to add a little bit of water. You don't want to just pour the water on the slide because you'll end up flooding it, and you have to wait for it to dry. So if you use too much water, you are going to be waiting all day for it to dry. So use as little water as possible. The way that you use as little water as possible is you put a loop full of water on. So over the sink, squeeze a little bit of water onto our sterile loop, just enough. Do you know how when you do bubbles, you can see that film of bubble soap there? That's all you need. Okay, then we're going to flame it again, because even though that's just water, it's not sterile water. Okay, now we have our next bacteria that's a solid that we're going to put onto our slide. Same procedure. Well, I'm going to wait for just a second. I don't think I'm quite cool. Okay, same procedure of pulling it out. Now that we're taking from a solid, we just want to barely touch the culture. Once again, little is more, okay? If you get a huge glob of it, you're going to end up with a huge glob. Once again, if you can barely see it, it's perfect. Don't gouge the agar. You want to be really careful not to bring agar with you onto your slide. Okay, now that's done. And the next thing that you're going to do is wait for it to dry. Now, I'm not going to stand here for 10 minutes waiting for it to dry. So I'm going to pretend that this is dry. As it dries, you'll start seeing the white part of the smear. We're going to allow it to dry, but like I said, I'm not going to wait. So you will wait, but I'm not. What you're going to do now is you're going to do what's called heat fixing. Some procedures have you fix with methyl alcohol. We will not do that. We will always heat fix. So once this is completely dry, meaning no water left, don't dry it by blowing on it or waving it in the air or anything. You're going to have to be patient. So you can make a bunch of slides at once, let them dry, work on the questions in your lab. When it's dry, then you can fix it. Okay, you have to be kind of a multitasker. So now that we're going to heat fix it because it's dry, even though it's not dry. So now we're just going to run it through the flames a couple of times. Watch out for your fingers. So if you put 2 of them on one slide, you're going to get kind of close to your fingers. That's why maybe at first you use 1 slide for each bacteria so you're not frying yourself. So you're just going to run it through a couple of times, just twice. That's all you need, and that's it. It's heat fixed. Okay? Now, the reason that you've heat fixed a slide and made a smear is because you're going to stain it. So remember, this is the procedure you will always go through before you ever stain a slide.